Alright everybody, welcome to my Exam 2 Essay 2 video. Okay, anyway, I'm going to have my notes with me and look through that a lot because there's a lot of shit in it we got to go through. So anyway, to start off, we're going to be talking about senses, kind of this whole video. And basically with our body, there's different receptors that detect different stimulus, as a result can make different action potentials. So things like burn or like heat, that'd be thermoreceptors. Pressure, vibrations, those are mechanoreceptors. Ligands would be chemoreceptors, and so on and so forth. Pain is going to be our nociceptors. So first off, we're going to be talking about taste and all of our different tastes and stimulus that cause that. So first, we're going to start here with sour. The stimulus for sour taste is hydrogen ions or hydrogen proteins. So essentially, on our tongue, we have all these different taste buds. You'll see them on like our tongue, all those different ridges and bumps. So on those taste buds, there's a different pocket of taste cells or taste receptors. There's about 125 of them per pocket. And so some of those taste receptors are going to be for sour. And so for sour to do its thing, we have right here, this is called an ENaC channel. Okay, and so what we're going to have is we're going to have hydrogen ions are going to come in, they're going to bind to this receptor here, and then what that's going to do is it's going to open up our ENaC channel, which allows more hydrogen ions to come in. And then with those hydrogen ions, it's going to kind of close up this potassium channel. You don't have to worry too much about that. Just know potassium isn't going to be able to get through or get out as much, which is going to result in quicker depolarization. And so that depolarization is going to come in and it's going to open up this voltage gated calcium channel. We're going to have all this calcium is going to flood in, going to bind to these snare proteins on this vesicle, and it's going to come in and it's going to kick out our neurotransmitter, which is going to be connected to a neuron that's going to synapse over to our brain. So that's sour. Pretty similar, if not, mostly similar, is salty. So salty taste, but our stimulus here is sodium ions. Okay, it's essentially the same thing. We have again here an ENaC channel, and then that ENaC channel, so sodium's going to come, it's going to bind, open up this ENaC channel, allow more sodium to rush in. Now there's no potassium channel that we're going to worry about, but sodium's going to rush in, come in, that depolarization is going to cause this voltage gated calcium channel to open, and calcium's going to rush into our cell, same thing, come bind to our vesicle, the snare proteins on that vesicle, and it's going to kick that vesicle out via exocytosis, and same thing, going to kick out that neurotransmitter, which is going to synapse with the neuron, which is going to then travel over to our brain. Okay, so that's salty and sour. Now, for our sweet, bitter, and umami, that's a bit different because those ones, rather than ENaC channels, it's going to use G proteins. Okay, so this is all the same. This is all for sweet, bitter, and umami, or savory. We're going to have different ligands or stimulus. So for sweet, it's going to be like carbs or sucrose. Umami, it's going to be things like glutamate. We're just going to put glut. Yeah. And then for like bitter, it's going to be something called quinones. Okay, so different stimuluses for those different tastes. But depending on what the taste and what stimulus it is, it's going to bind to that G protein, and what that G protein is going to do is once it kicks off that alpha subunit and causes a cascade, it's going to activate what's called phosphodiesterase C. Okay, and then what that's going to do is it's going to cause an IP3 pathway. So it's going to send IP3 down to our SR. So this is IP3 down to our SR, which has its calcium. Remember, our SR is fully calcium here. It's going to bind to that, which is then going to open up this voltage gated calcium channel, right? It's a calcium channel there, and allow calcium to flood out of our SR and then bind to our vesicle here again via exocytosis. It's going to kick that neurotransmitter out. Okay? So that's pretty much the difference with sweet, bitter, and umami is it's G protein regulated, and it uses an IP3 pathway with our calcium deposit in our SR, okay? So those are our tastes: sweet, bitter, umami, and salty. And then another thing would be like spicy. There's a chemical called capsaicin, which is in different like peppers and hot things. And that capsaicin, what it does is it activates nociceptors or pain, which makes it feel like your tongue is burning. And we measure capsaicin via something called the Scoville scale. So if it's high on the Scoville scale, it's spicier. It's got more capsaicin in it. If it's lower, it has less capsaicin, less spicy. That's what you need to know for that. And so one thing about taste, though, is it can be, I guess, amplified by smell. So let's move our board over here a little bit, just so we can have it more centered here. So I have smell. So this is essentially the dendrite on our smell neuron, I guess. And so pretty much if you went up your nose, you'd eventually hit top of your skull. I guess it'd be kind of the bottom of your skull. And there's something called the cribriform plate. And that's where the so that neuron is going to go through the cribriform plate into your brain. But this is essentially the dendrite of that neuron. So in top of your nose, that connects into your olfactory nerve, which then goes into your brain. So this is how it's going to detect it. So a very similar pathway here. And for our G protein, what's going to happen is it's going to have an odorant, which is just like a piece of what you're smelling. So that odorant, okay, it's going to come in, it's going to bind to that G protein, cause another kind of cascade where it kicks that alpha subunit off. We all know how G proteins are. But it's going to activate something called adenyl cyclase. Okay, and what adenyl cyclase does is it's similar to what our sweet, bitter, and umami did, but instead of an IP3, this is a CAMP pathway. Okay, and then what that CAMP is going to do is it's going to open this cation channel here. So it's non-specific cation channel. So we can have, we're just going to use sodium. So sodium can rush in, and that's going to depolarize our RMP here. So depolarization, that's going to hit this voltage-gated calcium channel. Okay. Now this voltage-gated calcium channel is going to release our calcium. Our calcium is going to be able to flood into the cell. I guess, no, sorry. Backspace here. Just kidding, calcium's coming in right here. Okay, so then calcium comes in and it's going to open up this voltage gated chlorine channel. So this is a non-specific cation, so sodium and calcium can rush into it, depolarize it, which is going to open up a voltage gated chloride channel, which is going to kick chloride out, which is going to depolarize it even more. And that depolarization is going to keep going to our olfactory bulb, down our olfactory nerve, and to our brain. Okay, so that's the gist of smell. Okay, put some markers down, flip through my notes really quick. Okay, so we've covered taste and smell, how those all work there. Now let's move on to vision. Excuse me for my drawing right here. I realize that's not the most. Anyway, so here's our eyeball right here. We have our cornea, which is just kind of that like little thin covering on top of your eye. And then our pupil is the hole. This is our lens. And then on our lens, we're going to have suspensory ligaments, which kind of help, I guess, adjust our lens. And around the lens and around the iris, we have what's called our ciliary muscles, okay? Now, that just kind of helps focus light is what's going to happen. Okay, so we're going to have light come in, going to hit our lens, and what we want it to do is we want all that light to go through and focus on this focus point on the back of our retina there, okay? But depending on how far away something is or how bright it is, our lens is going to need to adjust to bend that light so it's able to focus here. So what happens is our ciliary muscles are going to contract or relax. So if our ciliary muscles contract, they get tighter, smaller, it's going to cause these suspensory ligaments to relax or kind of get some slack. It's like if you take some string, you're holding it, ciliary muscles contract, that string is going to kind of form a loop. It's going to be laxed or slacked. And so what that's going to do is it's going to allow the lens to kind of go to its natural state. So it's going to thicken a little bit. So this lens is going to thicken, and what that's going to do is going to be a bit more convex, and so that's going to bend light in a little bit more, okay, onto our focal point. But say our ciliary muscles contract, okay, so you're going to contract, so what that's going to happen, or sorry, relax. It's going to tighten up those suspensory ligaments, which is going to tighten up our lens, which is going to cause it to flatten, which is going to be more convex, and it's going to cause light to bend out a little more, okay? Now let's see here, find my notes about nearsightedness and everything like that. Okay, so say if a person's nearsighted, what's going to happen is that light is going to be crossing and focusing in front of our retina. Okay, so nearsightedness means they can see really good up close, but not very good at a distance. So that focal point is in front of our retina here. So what we're going to want to do is we're going to want to bend the light out so that it focuses back at the back of the retina like we want it to. Okay, so you'd use like convex glasses here to help bend that light out so it can focus on the back of your retina. Okay. But say a person's farsighted or hyperopia, meaning they can see really good at a distance but not very good close up, that means your focus point is kind of happening at the back of your retina or behind it. So what we would need to do is bend the light in so that it can focus right where it needs to be. So then we'd use something called convex lenses to help bend that light back in. Okay? Anyway, at the back of your eye where your retina is, kind of the structure we're going to see is there's going to be like these black cells. Those are called pigmented epithelial cells. You don't have to worry too much about those. Essentially, they're just full of melanin, and they just help bleach your photoreceptors, your rods and your cones. Okay, and then kind of next to those, we're going to have our photoreceptors here. Get different markers and start to crap out. So we have our photoreceptors here. And like we were talking about how there's different receptors that detect different stimulus, a photoreceptor is a receptor that detects light and turns it into an action potential. Okay, so we have our photoreceptors here. Right above that is going to be a bipolar cell. And above that you're going to have a ganglion cell. What the ganglion cell does is it takes in all that information that's going to be relayed up, and it's all going to connect with a neuron, right? It's all just going to go straight to what's called your optic disc, which is where your optic nerve is going to kind of come in, and then that's going to be what's relaying the action potential over your brain. Okay. So now, if we're kind of take this part, and we're going to zoom in on it and see what exactly the pathway is for how that gets turned into an action potential here. So get it pulled up here. Okay. So let's start with dark. Okay. So if there's no light coming in here, this is another G protein here. And inside that G protein, there's something called 11-cis retinol. And so if that's dark, that 11-cis retinol is going to stay the same, and nothing's activating this G protein. So nothing's happening here, which means this little enzyme, this is called PDE, PDE is not going to get activated. And what PDE does is it's going to destroy this kind of gate here on our CGMP channel, which is the cation channel. It's going to close that. But since it's not activated, it's not able to close that. And what's going to happen is a bunch of these cations, like sodium, is going to be able to rush in, okay? And then what the sodium does, like it usually does, is it's going to depolarize, and it's going to open this voltage-gated calcium channel and allow calcium to come in. And then same as always, calcium is going to come in, it's going to bind to these snare proteins here, and it's going to release this vesicle. And inside this vesicle is a neurotransmitter called glutamate. Okay, and so glutamate's going to get released, and then this glutamate is going to bind to another G protein. Okay, this is your GLUR6 protein. Okay, so it's going to bind to this, it's going to cause this alpha subunit to kick off, and it's going to do something similar here, except this is called a TRME channel, which again, it's another cation channel. And so it's going to be able to close that. It's going to break it down a little bit and close that. So now no positive charges can get into our bipolar cell here. Okay, which means there's no depolarization, which means no voltage-gated calcium channels open, which means no calcium gets in, which means no glutamate. Glutamate is also in this bipolar cell. No glutamate gets released. And since no glutamate gets released, this AMPA channel on our ganglion cell doesn't get triggered. So then no action potential happens. So that's why when you're in the dark, nothing really happens, okay? It's kind of the same pathway. So let's get an eraser here. Now let's kind of go through it as if light was hitting this, okay? So let's kind of erase a few things here. All right, give me 1 second. Erase. No calcium's coming in. No sodium's coming in. Okay. My marker again. Fix this up a little bit. Okay. Now let's go through it as if light was coming in. Now one rule we got to remember is that light, it always causes less glutamate release, okay? Light causes less glutamate release. So what's going to happen is we're going to have some light, it's going to come in, it's going to hit our G protein. Remember that G protein has 11-cis retinol in it. I guess this whole thing kind of as a whole, this G protein is called opsin. Its alpha subunit is called transducin. But anyways, it's going to hit our opsin, hit that G protein, and it's going to change that 11-cis retinol. It's going to break that 11 bond. So it's going to turn into 11-trans retinol, which quickly gets like fixed back up and the bonds go back to normal. But that's what happens, and it causes that transducin subunit to kick off and activate our PDE. Remember what our PDE does is it closes this cation channel, this sodium channel. Does that have a special name? No. But it closes that so no cations can get in, no sodium can get in. And since no sodium can get in, there's no depolarization, and if there's no depolarization, then no calcium channels are opened, and no calcium is able to get in, which means no glutamate gets released, okay? So that's how light induces less glutamate, okay? But then what happens at our bipolar cell is now this G protein here, this GLUR6 protein, it doesn't get activated, which means its alpha unit doesn't kick off and close our TRME channel, which allows sodium or other cations to rush in because that didn't get closed, okay? And then what that's going to do is it's going to depolarize, open up this voltage-gated calcium channel, allow calcium in, which is going to cause the release of some glutamate. And then glutamate binds to an AMPA channel. An AMPA channel is always receptive to glutamate. So bind to that AMPA channel, which is is going to cause our action potential down our ganglion cell, down that neuron to our brain. So that's how light develops into an action potential, okay? So just remember the difference there between dark and light and how that all works, okay? Now let's get into a little more tricky stuff. This is what's called our receptor field. Now what happens with our receptor field, basically what a receptor field is there's different photoreceptors, different neurons that kind of do different things as far as action potentials go. So there's like a center ones in the center of the field, there's a center receptor, and then surrounding receptors called surrounding receptors, okay? And what that does is it helps us distinguish like shadows and light, so we can distinguish edges, so like where something's darker and where something's lighter. So we're gonna I'm gonna try my best to show how that works here. Now rule number 2 here is the receptors are named. We'll notice we'll use like on center, off surrounding, or off center, on surrounding. What that means is that's what's happening, is that essentially it's named after what it's doing to our ganglion cell here, okay? Okay, so let's go through it here. Okay, so essentially all these photoreceptors, so again it's our photoreceptors, bipolar cell, ganglion cell. So our photoreceptors, the pathways here is all the same. Everything's the same. One thing is we're going to add in some potassium channels and chloride channels here. And why we do that is because, as you can see, there's these 2 smaller squares. Those are called horizontal cells. Horizontal cells. Okay. Now what our horizontal cells have is another vesicle, but this vesicle has GABA. And GABA is an inhibitory neurotransmitter, so it's going to open up chloride channels, allow chloride to come into the cell, and potassium to leave, which is going to inhibit our center neurotransmitter here. Okay? And right here it also has an AMPA channel, so it needs glutamate to activate it. So if glutamate doesn't bind to this, then it's not going to inhibit our center one. Okay? And another thing is our on center, our on cell is essentially, a good way to think of it is it's got 6 glutamates, or 6 action potentials that it can activate, and our surroundings have 2 each, if we look at it like that. Okay? Alright, so now what we're going to do is we're going to essentially say, let's go, so we have 2 different options here, so let's start on center, off surrounding. Okay, so what that's going to mean is that we're going to have 6 action potentials come here because this is on. So it's activating 6 action potentials here. These are off, so their 2 doesn't come in. Now, let's say that light is going to come in, it's going to hit these 2, right? So essentially, we're going to have the same pathway we had here with the light. And rule number 1 is light always causes less glutamate to be released, okay? So essentially, no glutamate is going to be released, which means our horizontal cells aren't going to be activated or anything like that. They're not going to release GABA, not going to cause an inhibitory effect or an IPSP. So what's going to happen is really, so this is going to kind of turn off. That's going to turn on with light hitting it. So I have 6 action potentials are going to come in, and bam. So it's going to happen here. Going to go through that same pathway we did here. Glutamate gets activated, and now we're going to have 6 action potentials come through. It does the same pathway, except if we were to switch this to off, the only thing that would change is rather than a GluR6, we're going to have an AMPA right here. If this was off, this would be AMPA. Wouldn't have a TRME channel there. Okay. All right. Another thing to note is that it's named after kind of what light does. So on, off, basically light hits it, it's going to turn this off, and it's going to turn that on. But dark's going to do the opposite. Okay. So this one, it's off, which means it's dark. So we go through kind of our dark pathway, right? So we're gonna have that. Cation channel is gonna be open, sodium's gonna flush in. It's gonna open that up. It's gonna activate our glutamate. Gonna go through the same pathway as we did with that dark. Gonna release glutamate, and it's gonna activate this AMPA channel on our horizontal cell, which is gonna release GABA. And then that GABA is gonna inhibit that, okay? So then what that does, it's gonna release some of that, release some glutamate here. But remember, this is opposite. So the dark is actually turning this on, which is gonna result in 2 more action potentials coming through here. So I have 2 for that side. Very complicated, very hard to kind of teach via video, especially with limited time. So definitely go through that more and just understand how that works a little bit. But kind of the main rules is light releases less glutamate, and our receptor fields are named after what it does to our ganglion cell here. And then dark's gonna kind of do the opposite. And we only have 2 options here. We can have an on center off surrounding or an off center on surrounding, okay? Okay, and that's all there is for that, or at least all I can think of. So hopefully I don't get docked points there. I'm trying, guys. Just give me a break. Come on. Okay. Okay. Last sense that we're going to cover and then we'll be done is hearing. Now, hearing can also be a little bit complicated. So we're going to start here and kind of blow up different pieces of it. So this is our ear. So we have our outer ear here, right, which just funnels sound waves into our thing, okay? So we're going to send some sound waves down here. That's going to hit our eardrum or our tympanic membrane. And what that's going to do is it's going to be like a drum. So it's going to vibrate that. And what that's going to do is it's going to amplify our sound. So it's all going to compress, kind of amplify, and it's going to amplify on our malleolus. So then that's going to vibrate, which is going to vibrate our incus, which is then going to vibrate our stapes here. So our auditory ossicles, just those little bones in your ear. So sound's going to come in, it's going to hit that, and it's just going to vibrate that like crazy. So it's going to amplify those sound waves. And then it's going to kind of shake on what's called the oval window here, the stapes is. And then what that does is it enters those waves into our cochlea, which is like that snail-shaped thing there in our ear. Whoa, sorry. And our cochlea, we can kind of break it down into different sections here. So right here, this is going to be our, make sure I'm spelling this right again. Our scala vestibuli. And this is our scala tympani. And then this right here, this is our cochlear duct. And this blue line that I drew here, that's what's called our basilar membrane. So now we're going to go over here, where I essentially just took this basilar membrane and threw it out here. Now if we took our basilar membrane, remember our cochlea is kind of rolled up like a cinnamon roll. We're going to take that basilar membrane and we're going to unroll it. And if we were to unroll it, we would notice this, that it would be thinner towards this end here, and it'd be thicker towards here. This is like your helicotrohm. I think I said that right? Helicotrohm right here. It's going to be thicker there. What that does is it acts as like a violin or a guitar chord. So if we vibrate towards this other end, towards our oval window, that's going to result in high frequency, or like a high-pitched sound. If we vibrate it towards our helicotrohm, it's going to be low frequency or low-pitched, okay? So essentially our waves and that sound is going to come in and it's going to run down our basilar membrane, and depending on where those waves resonate on our basilar membrane is what sound we're gonna hear, okay? So now we're gonna try to explain how this can all get turned into action potentials for our brain. So what we're gonna do is we're gonna take our cochlea, and we're gonna cut it in half. We're gonna look at like a cross section of it right here. So again, right here, this is our scala vestibuli. This is our scala tympani here. And this is our cochlear duct. And we still have our basilar membrane here, the blue. Right here, this is going to be called our tectoral membrane. And then these purple things, those are what's called hair cells. And we're going to blow those up a little more too to show how those work. But starting here, our cochlea is just full of liquid, and these different sections have different liquids. So our scala, vestibuli, and tympani, it's filled with what's called perilymph. And perilymph, one thing you can know about it is it's high in sodium, okay? And then our cochlear duct, it's got what's called endolymph. And endolymph is high in potassium. Now with hearing, it's different than everything else. Usually sodium is what causes an action potential with our other senses and contractions and whatnot. With hearing though, it's potassium is what causes the action potential. And we'll explain that more when we open up these hair cells or zoom in on those. But the first thing we gotta know is we gotta get the endolymph high in potassium in order to cause those action potentials, okay? So we're going to take this section right here. This is what's called your stria vascularis. We're going to take that and zoom in on that, okay? So right here, this is this chunk, the sclera vascularis. We're going to show how we can get potassium from outside into our endolymph here, okay? So I have these different cells here on this edge. The first thing that's on them is our sodium-potassium pump. So we'll be pumping 3 sodiums out and 2 potassiums in. Let's go back to red. Red shows up better. Okay, so that's what's going on there. So then we're pumping in potassium here. Now, this is what's called our NKCC. Okay, our NKCC pump. So what that's doing is it's going to pump in a sodium, and it's also going to pump in potassium, and it's going to pump in 2 chlorides. We need that just so we don't have like differentiation in charges. So we're going to pump some chloride in there just to keep it neutral, so we don't have it too positive. So, but anyway, now we're pumping in even more potassium here. But then the sodium is going to be getting pumped out via the sodium-potassium pump. And then right here, this is a chloride leak channel. So then this chloride that just got pumped in is just going to leak right back out. Okay, so that's what's going on there. And it's going on for both sides here, both of these cells. So anyway, so now we're just pumping a bunch of potassium in. And then right here, we're going to have, forgot to draw them, but these are potassium leak channels. So now our potassium is going to come leak out in between that layer, okay? So we just took some potassium here, and now we just pumped it in and concentrated it even more here. But now we're going to go through the same process and concentrate it even more into our endolymph. It's the same process. We're going to have our sodium-potassium pump, pumping sodium out, pumping that potassium in. We're going to have our NKCC, just going to bring in some sodium, potassium, and our 2 chlorides. And those chlorides are going to go right back out via the chloride leak channels. And then our sodiums are getting pumped out. And then now we've concentrated it even more. So where now we have a very high concentration of potassium in our endolymph. Okay. Now, let's go back to this here, to our cross section here. what's gonna happen is, remember those sound waves, those pressure waves are vibrating our basilar membrane here. And so we've got a lot of movement on this basilar membrane. And what that's gonna do is it's gonna bump these hair cells into our tectoral membrane. So it's gonna be hitting that tectoral membrane with those hair cells, okay? At least our outer hair cells, but we'll get into that a little more. So now, the conditions are right, we're getting sound into our ear, our endolymph is high in potassium because of what we did here. Now we can use this potassium in the endolymph to result into an action potential in these hair cells. So now let's go to our hair cells here. Remember what we said, the basilar membrane is going to be vibrating and moving, and so it's going to hit that against this our tectoral membrane. And then what that does is it causes these little cilia-like structures to kind of bend out like that. And what we have here is we have, let's not draw it right there. These channels here, these are mechanically gated potassium channels, okay? So we need some sort of mechanic stimulus to open those up. So when those hair cells bend from when they're hitting the tectoral membrane, it has these little like mechanical structures. I've heard them called tip links. But what it's going to do is it's going to pull on it and it's going to open up that potassium channels there, okay? So now we're going to have all this potassium in our endolymph, and its driving force is just going to be so incredibly high that it's just going to rush right in. We have some potassium leak channels here too, so we're going to leak that out. We also have a sodium potassium pump, which is going to keep the potassium high in our hair cells, but also keep our perilymph high in sodium. But our driving force is going to be so much greater up here that potassium is just going to rush right into our hair cell there. So then what that's going to do is it's going to cause a depolarization. It's going to cause an action potential. It's going to open up this voltage-gated calcium channel. So now calcium is going to rush in, again bind to our snare proteins on this neurotransmitter, or this vesicle that has a neurotransmitter. Don't know what the neurotransmitter is, but it doesn't matter. That's going to kick that all out. It's going to synapse with this neuron here, which is going to send that signal to the brain saying, hey, this is the sound we're hearing now, okay? Now, this would be what's like our inner hair cell. So it's going to be like this dude here, which is going to regulate hearing, or it's going to detect the hearing. These inner one, or these outer hair cells, do what's called regulation, okay? So that's what causes more of our like selective hearing. So if we're at like a party and there's a lot of noises going on, our brain, via an efferent neuron, so these neurons here are efferent, can tell these hair cells and kind of regulate it a little bit and tell it like, hey, don't like chill out. You don't need to hear that right now, and it won't hear that. But we notice this like inner, so that's what's going on here a little bit. I guess with both, this is what's kind of going on. But with these outer cells, or I guess both of them too, we have a protein called Prestin. And what Prestin does is it acts as like a spring. So Prestin used to be like a voltage-gated chloride channel, but now it's broken, so now it's just a spring. And allows our hair cells to kind of do this. So as this basilar membrane's going crazy, those hair cells can also move. And it can say, oh, we're not going to move as much, or it can pull this about. And so it can kind of regulate our sound even more that way, and kind of tell us what we can hear, or what we should hear or not. Our brain also like changes up voltages too, but we don't need to worry about that too much. But just remember, Prestin acts as like a spring. And with that extra motion, it causes like kind of a wave in our perilymph, which is going to bend these hair cells, cilia, even more, which can cause this whole thing again and cause more action potential for that sound there. Okay, but that's everything as far as sentences go. Hope you enjoyed it. Please give me a good grade. Thank you very much.